This protocol provides a potentially standard pipeline of analyzing interbrain synchronization in fNIRS Hyperscanning studies. The main advantage of this technique is that it allows calculating interbrain synchronization via wavelet transform coherence method and validating interbrain synchronization with permutation-based random pairing of traps, conditions, and participants.
Perform all data analysis using the MATLAB software with appropriate toolboxes. During data preprocessing, specifically using the principal component analyses and correlation-based signal improvement method, remove the fNIRS global physiological noise using the NPCA filter and the head motion artifacts using the HMR motion correct _CBSI of Homer 2. Next, for calculating interbrain synchronization, or IBS, adopt the wavelet transform coherence function of cross-wavelet and wavelet coherence toolbox with default parameters, and compute the coherence values at each time and frequency point to obtain a two-axis matrix of coherence values. For the default parameters, use the Morlet mother wavelet to transform each time series into the time and frequency domain by the continuous wavelet transformation. Next, to choose the frequency band of interest, select and average the coherence values of the frequency band between 0.5 to 1 Hz according to the frequency band used in the finger motion task of the fNIRS Hyperscanning study. Obtain one column of coherence values for each pair. Then, select